In this section, I'm going to talk about the internal prokaryotic cell structures. So first we're going to start with the genetic material of a prokaryotic cell. The bacterial chromosome is a single circular strand of DNA. This isn't the case for all bacteria, but a majority of bacteria have one single circular strand of DNA. It's aggregated in a dense area called the nucleoid. So it's not a nucleus, it's not enclosed by a membrane, but it's just in a region that's dense and it's called the nucleoid. DNA is tightly coiled around basic protein molecules to fit into the cell compartment, and those proteins are histone-like proteins, but they are not true histones. Plasmids are non-essential pieces of DNA. They're separate double-stranded circles of DNA. They're duplicated and passed on to offspring during replication, and they usually have extra genes that have some type of protective trait or some ability to digest a certain nutrient. They're an important agent in genetic engineering, and we'll talk much more about plasmids once we get to genetics. So with bacteria, they typically have their one single huge chromosome, and then they have these tiny little plasmids, which are very small circular pieces of DNA that contain extra genes. Those genes are usually beneficial, but they're not essential for the survival of the species. Ribosomes. So ribosomes make protein. They're made up of RNA and protein. That's ribosomal RNA is what constitutes a ribosome. And the bacterial ribosome is smaller than the eukaryotic ribosome. Inclusion bodies store nutrients for the bacterial cell. They vary in size, number, and content. Some of them contain carbon molecules, and that's going to be a carbon storage site. Other ones contain metallic granules that are going to help them move through swamps and such. So different types of things can be stored there. They're kind of like equivalent to the vacuole of a eukaryotic cell. However, they are not enclosed by a membrane. It's just a pile of stuff in the center here. If we compare a eukaryotic cell to a 3-bedroom apartment and a prokaryotic cell to a studio apartment, you think of the eukaryotic cell, it has a separate compartment for its nucleus, so we can call that like the library inside of the 3-bedroom apartment. It has a separate compartment for energy production called the mitochondria, so we can call that the fitness room where energy happens. And then it has a separate compartment for storing nutrients. We can call that the food pantry inside of the apartment. But when we think about a studio apartment, it's really just a room. And in one part of the room, there's a bed. Then there may be like a pile of laundry somewhere. And then there's like a kitchen area. But nothing's really enclosed off by walls. It's all kind of open in one room. And that's what you can find on the inside of a prokaryotic cell. There's no membranes around anything. There's just areas of nutrients or areas of genetic material piled up, regions where a lot of energy can be produced. So it's not enclosed with a membrane. It's just an area in the cell where something happens to be. The cytoskeleton. So the cytoskeleton, long polymers of proteins that are similar to eukaryotic actin. They're arranged in helical ribbons around the cell just under the cytoplasmic membrane, and they help contribute to cell shape. They are a potential target for antibiotic development. So now we're going to talk a little bit about endospores. So previously I talked about biofilms and how dangerous biofilms can be. They're aggregations of multiple species of microorganisms living on a surface. A lot of times those will live on like your teeth or on indwelling devices in the body. If people have like heart machines hooked up or respiratory equipment, respirators, catheters, urinary catheters, biofilms can form there and they're really tough. So biofilms are one of the major struggles in the healthcare field. But another major struggle in the healthcare field is endospores. Endospores you can think of as bacterial seeds. They're dormant forms of the bacteria that are extremely resistant to environmental conditions. Only certain bacteria can make endospores, but when they produce endospores, those endospores can survive millions of years on inanimate surfaces. And millions of years later, They can take an endospore and put it in the correct environment, and it'll hatch open and become a brand new bacterial cell, a living, breathing, moving bacterial cell that's viable. And they've done this. They've regenerated million-year-old endospores and created living, moving, respirating bacterial cells. So this is something that's been done, that we've found these endospores from a long time ago, and they're still able to come to life. So endospores withstand hostile conditions and facilitate survival. You can spray disinfectants all over endospores. You can hit them with UV light, and they're resistant to that. They're resistant to pretty high temperatures as well. The only thing that can completely destroy an endospore would be sterilization methods. So really, really harsh chemicals, or an autoclave machine could do it. So an autoclave uses pressurized steam in order to completely sterilize something. But endospores are really tough to get rid of. They have the vegetative state, that's the living, breathing, moving state. And then they have the endospore state, which is an inert resting condition, ready to hatch open when the environment is correct. Sporulation is the spore formation that's induced by environmental conditions. So if a patient comes in with, for example, C. diff. C. diff is a diarrheal infection. Diarrhea tends to splatter, and sometimes these the bacterial cells can land on a toilet seat, for example. So let's say that bacterial cell is sitting on a toilet seat, and it starts to dry out, and it experiences that the environment around it is no longer the comfortable, nutrient-rich colon of a host organism. Now it can sense that there's no nutrients, it can sense that it's drying out, so the bacterial cell is going to quickly copy its genetic material and start to produce an endospore. The bacterial cell itself is going to die off, but that endospore is left behind from the inside of that bacterial cell. Now that endospore can stand there forever, and that endospore can be touched by someone else, and that's how those types of infections can be transmitted. So sporulation is just the process of forming the endospore, and that's dependent on environmental conditions. Like I said, if that bacterial cell gets into an environment where there's no nutrients, it's going to start forming spores. Endospores can resist heating, drying, freezing, radiation, and chemicals. The stimulus for the endospore formation, if there's a depletion of nutrients, especially carbon and nitrogen sources, the bacterium is going to produce an endospore. The sporangium is the sporulating cell, so it's the cell that's actually producing the spore, and the transformation takes 6 to 8 hours in most species. Some diseases are related to the persistence and resistance of their spores. So Bacillus anthracis, Clostridium tetani, Clostridium perfringens, Clostridium botulinum, those cause some really wicked infections. So anthrax, tetanus, gas gangrene, botulism, and most often it's the spores that are causing the infection. So the spores are getting into the body. The spores are the ones that are being transmitted from one organism to the next, or contracted from the environment, and that's what's making us sick. Medical significance of endospores, because they can constantly be on surfaces, they're always an issue in the medical field. We don't know that they're there. We can't see them. And even if we spray disinfectant all over the place, we're still not getting rid of the endospores. So they can persist for a really long time. They cause a lot of really severe infections. So not only are they resistant, but the infections that they cause are really severe, and they can cause death in many cases. They frequently contaminate cultures and media. Hospitals must protect against endospores in wounds. And destruction of endospores is important in the food canning industry. So we'll learn more about Clostridium botulinum later on in the semester.